Human reproduction is a sexually dimorphic system, requiring the fusion of a male and female gamete. Because humans are viviparous, the resulting embryo develops entirely inside the maternal body before being born alive. The process functions as a strictly chronological biological pipeline, where every stage relies on the successful execution of the step before it. The process moves through distinct macro stages: the initial formation of gametes, their physical transport, fertilization, the implantation of the embryo into the uterine wall, and finally parturition, or childbirth. Yet this entire machinery remains completely dormant throughout childhood. It only activates at puberty, triggered by highly specific neuroendocrine signals originating in the hypothalamus. From that moment on, human reproduction operates through an unbroken chain of chemical and physical handoffs between massive anatomical structures and single microscopic cells. We begin the mapping of this pipeline in the male pelvic region. The primary sex organs are the testes. They are responsible for a dual output, producing the male gametes, sperms, and synthesizing male hormones, primarily androgens like testosterone. This simplified thermal scale compares core body heat to the strict requirements of sperm production. Spermatogenesis needs a temperature exactly 2 to 2.5 degrees Celsius lower than the rest of the body. To maintain this, the testes are located externally, suspended in the scrotum. Inside, each testes is densely compartmentalized into about 250 testicular lobules. Within these lobules lie the highly coiled seminiferous tubules, the actual sites of cellular generation. Once produced, the gametes travel through a sequence of accessory ducts. They move from the tubules into the epididymis, where they are stored and mature, and eventually route through the vas deferens to exit the body via the urethra. The male reproductive system functions as an externalized, temperature-controlled manufacturing plant, optimized for a continuous, high-volume biological output. The female reproductive anatomy operates on a completely different set of biological constraints. The primary organs, the ovaries, are located deep internally, anchored securely to the pelvic wall and the uterus. Looking at this cross-section, we trace the transport corridor. An unreached ovum is swept into the fallopian tube by the funnel-shaped infundibulum and its fimbriae. It then moves into the wider ampulla. If fertilization occurs, it must happen exactly here. The final destination is the uterus, an inverted pear-shaped organ. Its walls are heavily stratified, featuring a tough outer perimetrium, a thick muscular myometrium, and a highly active glandular inner lining called the endometrium. Rather than continuous high-volume production, the female anatomy is an internal, cyclical incubator designed to capture, transport, and nurture a single highly valuable cell. Supplying these anatomical systems requires intense cellular division. In males, this process, spermatogenesis, begins at puberty inside the seminiferous tubules. This cellular division flowchart begins with diploid spermatogonia. Containing 46 chromosomes, these cells undergo mitosis, differentiating into primary spermatocytes. The first major transformation is meiosis I. This reductive division splits the primary spermatocyte into 2 secondary spermatocytes, reducing the chromosome count from 46 to 23. A second division, meiosis II, follows immediately, yielding independent haploid cells known as spermatids. These static spermatids then undergo a structural transformation called spermiogenesis to become fully functional spermatozoa. Once mature, they are released from the tubules in a process termed spermiation. This schematic details the final anatomy of the spermatozoon. The head carries the genetic nucleus and is capped by the acrosome, which stores enzymes necessary for fertilization. The middle piece is densely packed with mitochondria to generate energy, powering the tail for rapid motility. This entire assembly line is driven by hormones from the pituitary gland. Luteinizing hormone stimulates the Leydig cells to produce testosterone, while follicle-stimulating hormone acts on the Sertoli cells to drive spermiogenesis. Spermatogenesis operates as a continuous, exponential replication machine, churning out millions of highly mobile, energy-dense genetic delivery vehicles every day. Oogenesis follows a different timing strategy than the male system. The entire supply of female gamete mother cells, oogonia, is generated during embryonic development, meaning no new oogonia are formed after birth. These cells develop into primary oocytes, but their maturation immediately halts. They are arrested in the prophase 1 stage of cellular division, sitting dormant for years until the onset of puberty. Once puberty begins, the maturation pipeline restarts in a cyclic pattern. A primary follicle slowly develops, accumulating surrounding granulosa cells until it reaches maturity as a Graafian follicle. The payload inside this mature Graafian follicle is a single secondary oocyte, wrapped in a protective non-cellular layer called the zona pellucida. Oogenesis is a highly conserved, front-loaded biological strategy that meticulously matures only a single viable gamete per cycle. To coordinate the release of that single egg with the internal environment of the uterus, the female body utilizes an average 28-day hormonal rhythm known as the menstrual cycle. This 28-day line graph tracks the follicular phase. As FSH and LH drive ovarian follicle growth, rising estrogen forces the uterine endometrium to rapidly thicken. Around day 14, a sharp LH surge physically ruptures the Graafian follicle. This singular event is ovulation. Following ovulation, the cycle enters the luteal phase. The ruptured follicle transforms into a temporary endocrine structure called the corpus luteum. The corpus luteum produces immense amounts of progesterone. This hormone acts as a chemical pillar, maintaining the thick, nutrient-rich endometrial wall in preparation for a pregnancy. If fertilization fails to occur, the corpus luteum degenerates. Progesterone levels collapse, the unsupported endometrium breaks down, and bleeding marks the start of a new cycle. The menstrual cycle is a precisely synchronized biological clock that ensures the release of an egg perfectly aligns with the architectural readiness of the uterus. When semen enters the vagina during coitus, millions of sperm utilize seminal plasma to survive the transit through the cervix and uterus, racing toward the solitary ovum waiting in the ampulla of the fallopian tube. Enzymes within the sperm's acrosome dissolve the ovum's outer barriers. The head penetrates the ovum, allowing the genetic material of the two gametes to fuse. The instant this fusion occurs, a biological lockdown is triggered. The zona pellucida chemically alters its structure, completely blocking the entry of any additional sperm to prevent polyspermy. This fusion creates a diploid zygote and instantly determines biological sex. Because the ovum always carries an X chromosome, the paternal payload dictates the outcome. An X-carrying sperm results in an XX female, while a Y-carrying sperm results in an XY male. As the zygote begins its journey down the fallopian tube, it undergoes rapid mitotic divisions called cleavage. It splits into 2, 4, 8, and 16 cells called blastomeres, forming a solid morula, which further hollows out into a structure known as a blastocyst. Fertilization is an immediate genetic catalyst, instantly converting two highly specialized haploid cells into a rapidly dividing self-organizing structure. Upon reaching the uterus, the blastocyst embeds itself deep into the prepared endometrium. This physical attachment is implantation, marking the official beginning of pregnancy. To survive, the embryo develops finger-like projections called chorionic villi. These interlock with maternal uterine tissue to construct a shared joint organ, the placenta. Connected by the umbilical cord, the placenta is a biological bridge supplying maternal oxygen and nutrients while removing fetal waste. It also operates as an endocrine engine, producing essential hormones like HCG and HPL to sustain the pregnancy. Supported by this constant chemical pipeline, embryonic development moves rapidly. By the end of the first month, a functional heart forms. Limbs develop by month 2, and by 12 weeks, all major organs are structured from 3 distinct cellular germ layers. The placenta functions as an external life support system, seamlessly linking the maternal anatomy to the demanding metabolic needs of the growing fetus. After approximately 9 months of gestation, the pipeline culminates in parturition, or childbirth. The biological trigger for this event originates entirely from the fully developed fetus and the placenta, generating the fetal ejection reflex. This flowchart outlines a self-amplifying positive feedback loop. Initial fetal signals cause mild uterine contractions. These contractions stimulate the maternal pituitary gland to release the hormone oxytocin. Rising oxytocin levels force progressively stronger contractions in the thick myometrium layer of the uterus. These stronger contractions trigger even more oxytocin, escalating until the baby is successfully delivered. Following delivery, the mammary glands activate to produce milk. The initial secretion, called colostrum, is packed with pre-built maternal antibodies, delivering immediate, vital immunity to the newborn. Human reproduction operates as a precise biological pipeline. It relies on neuroendocrine signals, specialized cellular divisions, and anatomical synchronization, ensuring the successful formation and independent survival of a new human life.